genetic material in the DNA. Genetic material in the DNA means the character controlling alleles are there in the DNA. If the character controlling alleles are there in the DNA, if the character controlling alleles are there in the DNA, okay, we are calling the DNA as a genetic material, right? So now we are coming for the next point is in the search for the genetic material. What we need to understand is the genetic material is the DNA, which is carrying the message from one generation to other generation. We know very well our Gregorian Mendel, okay, Alfred Sutton, and then Thomas and Morgan established the genes are located on the chromosome, okay, in a cell nucleus. In a chromosome means exactly you need to understand in a chromosome the character controlling alleles are there in a specific location. Character controlling alleles are there in a specific location. So character controlling alleles are there in a specific location denotes what? Locus. Locus is singular. Loci is plural. Again back I'm repeating, loci is plural. Locus is singular. In the locus, basically the character controlling alleles are present. Character controlling alleles are present in the locus. Right? Character controlling alleles are there in the locus. Right? Now, in this Frederick Griffith experiment, what we need to understand, Streptococcus pneumonia is a bacteria which causes pneumonia disease. In this particular disease, what happens exactly? We are understanding the problem in the transformation. The problem in the transformation of one particular species is not because of the protein, it is because of the DNA. Like this, we are understanding. Calmly you listen. The strain is also called a smooth strain. If you are injecting into the rat will die. Whereas in the case of heat killed, we are killing the strain. What do you mean about strain? Smooth strain. What do you mean about smooth strain? Shiny. What do you mean about shiny? It is having polysaccharide. What do you mean about polysaccharide? Carbohydrate. What polysaccharide? It is exactly made up of many monosaccharide. So that polysaccharide made up of many monosaccharide, and that too exclusively a different type of monosaccharide, is responsible for giving the changes in the membrane, right? Calmly you understand again back what is called S cell, smooth strain. Why it is called smooth strain? Why? Because it is having the lipopolysaccharide. So what is in by R strain, rough strain? It is not having the lipopolysaccharide. It is not having lipopolysaccharide. So R cell has lipopolysaccharide. S strain is not having lipopolysaccharide. When we are injecting the lipopolysaccharide or lipopolysaccharide rich microorganism, the rat will die or mouse will die. Okay, whereas in the second case, after killing the microorganism, the mouse is not dying. Okay, if you are injecting the R strain means R strain is not having a lipid carbohydrate or lipopolysaccharide in the membrane to get adhered, to get attached to the host cell. Okay, for that the R cell is not having. There is a reason why, even though the pneumonia of R variety, if it is entering into the mouse, nothing will happen. Mouse is alive, happy, everything is okay. But if you're mixing the R strain along with the S strain, what happens is here exactly mouse will die. Mouse will die. So this is the way we need to keep in our mind how the chances of getting infection. Okay, the chances of infection, or else you can say the infection probability in a one particular species because of this particular variety. In microorganism itself, there are totally two important varieties are there: R strain, S strain. The S strain basically having lipopolysaccharide, R strain not having lipopolysaccharide. If you are not having lipopolysaccharide, being R strain, then you are not having credential to attack the host cell, right? So what happened is the S strain and the R strain we mixed. So the R strain and S strain we mixed. So this S strain is killed. Okay, the killed S strain and then alive R strain are mixed, and then we injected into the mouse, and then we found out the mouse is dead because the reason because the R strain became the S strain. Though the R strain became the S strain. Okay, how the R strain became S strain? Because the DNA of R strain is transformed. Okay, the DNA of the R strain is influenced by the S strain. It's influenced by the S strain, right? So now we are coming for the next important point is Streptococcus pneumonia. Streptococcus pneumonia is a bacteria, okay, which is causing a lot of issues in the human being as well as in the mouse, right? So now we are entering into the next experiment. Frederick Griff transformation experiment denotes DNA is responsible for bringing the transformation. Okay, for bringing the transformation of which species or strain to what? Yes, strain, right? So because of that, what happened? Mouse died. Why? Because mouse cell are attacked by this S strain. Okay. Now we are coming for the next point is the transformation principle, which is furthermore narrated by Avery, MacLeod, and McCarthy, are the two great scientists. Instead of saying like this, three great scientists determine the biochemical nature. The biochemical nature, what it is exactly, what is the conclusion of this? So the R strain DNA is transformed into the S strain DNA because of the entry of the S strain DNA into R strain. So the R strain is transformed into S strain after the entry of the S strain Streptococcus pneumonia into the R strain body. Okay, so again back I'm repeating into the pneumococcus body. Okay, so who entered means Streptococcus entered. Streptococcus entered, right? So R strain become S strain. R strain become S strain. So now we are entering into the Archie Chase experiment, which is a bacteriophage experiment. If you're calm, quiet, and listening to the concept very properly, then you can understand. Uh, Here, this picture explains multiple information. So what we are going to understand from this experiment is the DNA is responsible for transforming the other species. Okay, but RNA also responsible in few species like HIV. But in most of the species, one species is getting transformed into other species only because of the DNA, right? So you know very well this bacteriophage after getting attachment to the bacteria. This bacteriophage after getting attachment to the bacteria. So what it does exactly? This bacteriophage injects the DNA, injects the DNA. So in the first case, we grown the bacteriophage in a nutrition medium having the radio labeled sulfur we used to call as 35 years. This 35 years is nothing but a light radioactive isotope. This 35 years is nothing but a light radioactive isotope, right? So again back I'm repeating this 35 years to the upper script. At the left hand side we are writing 35 years and 32p to the upper script, the left hand side you are writing. So in the left hand side of you can see this 35 and 32 denotes light radioactive isotopes. I'm growing one bacteriophage in 35 years, another bacteriophage in the 32p, right? So what we need to understand basically is the when you're growing this two bacteriophage, the two different nutrition medium, one is sulfur, another is phosphorus. Sulfur is used for making the protein. That protein is not entering the protein or the bacteriophage is not entering into the bacteria, whereas the DNA, which is radio labeled, okay, which is entering into the bacteria and made the bacteria to become radioactive. Okay, here you need to understand basically. So in this bacteria, there is no chance for again I'm repeating in this bacteria there is no chance for radioactivity. So there is no radioactivity in this case. Radioactivity is there. So why? Because the radioactivity DNA entered into the bacteria, and then when we are exposing this particular bacteria, wherever I given the trick to the X-ray film, we are finding the positive result. What is positive result? You are saying, Hari sir. So when we are exposing this bacteria to the X-ray film, what happens exactly? Can you tell me? Even though my biology teacher of CBSE board not told, this radioactive radiations converts the silver nitride to silver nitrate. As a result, in the X-ray film, you can see the spots over it. This spots is directly proportional to the amount of radioactivity, right? So this sentence is missing in the CBSE textbook. So that is why we are having the control. We are not going behind certain limit out of the CBSE board, right? Whatever the CBSE board has given, see in that only what we are explaining all the concept in detail, right? Anybody are having any doubt in this junction? Okay, in the green, we are observing the radioactivity. In the left-hand side, the brownish one, we are not observing the radioactivity. Why? Because the transformed one is only because of the DNA. The DNA is transformed in the bacteria because of the phage. The phage is having this radio-labeled DNA because it is grown in a nutritional medium having radioactive phosphorus. Radioactive phosphorus. Again, back I'm repeating. Most of our students are writing 32p like this. If you're writing like this, you will be not getting the mark. Then how I need to write? I already told you superscript. Okay, subscript, you know very well. And that too in the left-hand side, you need to write this particular symbol information, right? So now we are entering into the next concept, which is nothing but in the next to the bacteriophage experiment, right? We are entering into the properties of genetic material. Before entering into the properties of genetic material, what we'll do is we'll go to certain examples. Okay, this is a worked example which makes you to understand the significance of every McLeod MC-Corti experiment in determining the nature of the genetic material. Determining the nature of genetic material. Right on. So what you need to understand basically is so this experiment, how we narrated in terms of answer script while writing the exam. Okay, how we need to narrate that we already indicated for you. This is the way you need to write the information. This concept we already discussed just now only. Okay, and the heat killed a strain or strain, everything we discussed, right? If it is 2 mark, sentence is enough. If it is 3 mark, sentence is enough. If it is 5 mark, compulsory, I need to draw the diagram. The diagram we already explained for you, right? So now, this is one of the worked example. Okay, other worked examples we'll discuss furthermore in detail in the other subdivision. Before that, we'll go for tips for exam. In the tips for the exam, what I need to study, how I need to map my brain in such a way I will get maximum score. For that to happen, we need to have the historical information, pros and cons. This is the pros, this is the cons. Okay, this is the plus, this is the minus. So because of that, who is the next scientist brought the advancement, and that advancement impact over the research field in a very precise way we need to narrate. Then only what the time management is possible. Or else if you're keeping on narrating within the time period, we can't finish it, right? So time management is very important. Number two, for 2 mark, how much you need to write? That much you need to write. Okay, for 5 mark, the compulsory diagram like this, what you do for this particular Frederick Griffith transformation experiment, right? Be very familiar with the technical terms. In the Frederick Griffith experiment, what are all the technical terms we discussed? Transformation, right? Then what are the other things we discussed? The estrain is because of the lipopolysaccharide existence, unlike orstrain. Why it is called R? Why it is called S? Okay, why it is called R? Why it is called S in the case of the bacteriophage? Like this, if they're asking, the R is because of this and S is because of this, like this we need to say, right? So this R and S, everybody are sleeping very nicely. This R and S we not used for the bacteriophage. We used only for the bacteria, which is your Streptococcus pneumonia, just only we discussed, right? So here this every experiment, what it speaks about, right? Frederick Griffith experiment, what it speaks about. After Chase experiment speaks about bacteriophage. So some students are writing bacteria, Streptococcus pneumonia in the case of Harsha Chase experiment. Some are writing this radio labeled activity experiment in the case of Frederick Griffith experiment. Both are wrong. So specific microorganism for a specific experiment and outcome of the experiment and how they did everything we need to narrate in a periodical way, right? So now we are coming for the next important segment. Whenever you're having time, have a look into the related video we posted for you for better understanding. Now we are entering into the next important sub topic is nothing but properties of the genetic material, DNA versus RNA. Once upon a time, I already told RNA is a genetic material. Once upon a time, what I told you, RNA is a genetic material ruling the world more dominantly in most of the species, but not in all species. But not in all species, right? Be very careful with all these things. Okay, again, back I'm repeating. In few species, only in few species, okay, the DNA existed as a genetic material, whereas in most of the species, RNA existed as a genetic material, right? Why the RNA, okay, got transformed? The reason behind that is it is having the free hydroxyl group in the second carbon atom that we already told to you before 30 minutes back, right? So because of the free hydroxyl group in the second carbon atom, because of the free hydroxyl group of the second carbon atom, what happens is exactly the RNA is highly prone for getting mutated to DNA. Okay, so this is the way most of the RNA genetic material become DNA genetic material, right? In the current trend, in the current era, so most of the genetic materials are DNA, whereas few genetic materials are RNA. Of course, we are accepting it. Of course, we are accepting it, right? So now we are coming for the next phase explanation in the genetic material. We know very well the presence of thymine. The presence of thymine instead of uracil in the RNA. Thymine impacts the additional stability of the DNA. So the stability of the DNA, okay, means what exactly? I am not prone for getting mutated very faster, right? RNA mutates at a faster rate due to instability. Why? Because it is having free hydroxyl group in the second carbon atom. So this we know very well in the chemistry. We already studied functional group carbon. Okay, functional group carbon are highly prone for getting mutated. Okay, but here this hydroxyl group is not a functional group carbon. It is having a free hydroxyl group, which is having the potential to interact with acid or alkali very faster. So this acid alkali or acid or base, when it is interacting with the particular free hydroxyl group, the RNA is becoming DNA, right? So now we need to understand the DNA is preferred for a long-term storage of the genetic information due to the greater stability, right? So what are the information we are observing in the superior breed because of the DNA is transformed very instantly like this? We can't say. Okay, why? Because it is too difficult for the DNA to become other DNA. Okay, it takes millions of years to become other DNA. But if we are doing artificial mutation, for example, our DNA is getting exposed to the very tough atomic bomb blast, or else if it is getting exposed to the tough Bhopal tragedy yesterday we told to you, or else in the bioweapon applications, the DNA will become other DNA within a short period, but not for the natural process. This concept we can take it. Yeah, there is a reason why I'm saying the short DNA. The short DNA will become the other DNA in few years if you're doing the mutation continuously. But even the long DNA takes a lot of time to get mutated, and then it changes the gene sequence appropriately, and then it gives the proper information. Now we are coming for the next phase. See, in the expression of the character, so I already told you one is short DNA, another is long DNA. Long DNA is highly prone. No, only short DNA is highly prone. But short DNA takes less number of months, whereas long DNA takes many years. And then again back in the natural process, it takes many years to go. Whereas in the case of induced mutation in experiment, it takes only few years, right? Now, in the genetic material criteria, the DNA satisfies this criteria more effectively, right? So what we need to understand is the DNA on the other hand relies on the RNA as an intermediate for the RNA protein synthesis, right? So as we told in the yesterday's session in the genetic disorder, the whatever the changes which is happening in the cell because of the change in the change in the protein is because of the change in the RNA. The change in the RNA is because of the DNA. So like this we already told you, right? So I told about DNA, I told about RNA, and then I told about protein, right? So now we are coming for the next phase explanation, the RNA virus. What do you mean about RNA virus? So we know very well tobacco mosaic virus is nothing but a RNA virus and QB bacteriophage. Like this, many RNA viruses are there. These RNA virus basically responsible for causing the mutation, and then second thing is it also have the credential to undergo mutation. Sir, you are saying it is causing mutation, and then it is undergoing mutation. Which one I need to take? Take both. Can you give an example? Yes, of course, HIV. Okay, how it does exactly? The HIV RNA undergoes changes on its own. It also causes changes in human DNA, right? So it works on either side. So there is a reason why we told the dual behavior sense. For example, the light is having particle property and wave property. Okay, the dual behavior sense. In the same way, if you're talking about the virus, it's also having the dual behavior sense in terms of changing the other DNA sequence and RNA sequence that is agreed only for the RNA virus. That is agreed only for the RNA virus, but not for the DNA virus, right? So what we need to understand is the genetic expression and protein synthesis. Okay, RNA plays a crucial role in the genetic expression and protein synthesis, but it is more reactive and subject to rapid mutation, right? So the reason is free hydroxyl group that we already narrated for you, right? So now we are coming for the next segment. Before going to the RNA world, so what we'll do is we'll go for the examples, worked examples, and then we'll go for some few questions. Compare the stability of the DNA and RNA and explain why the DNA is preferred as genetic material, right? So because whatever the superior breed information which is there in the DNA, which won't be transferred to the other generation, which won't be transferred to the other generation if there is a mutation. If there is a mutation. Whereas in the case of the RNA, okay, it is highly vulnerable for getting changes. Okay, so simple logic is the DNA is highly prone for getting mutated. No. RNA is highly prone for getting mutated. Yes. And second thing is DNA also prone for getting mutated if it is exposed to the mutagenic agent. Okay. So we need to understand the pros, cons, conditions, etc. Then we need to narrate the answer in detail. For example, in which occasion the DNA is highly prone for getting mutated than the RNA. So we can say, yes, of course, that possibility is there if the mutation is exposed to the DNA by a strong mutagenic agent. For that, we already had given the number of examples yesterday, right? So these are all the classic narration for the worked examples, which is nothing but the hot question, exclusively prepared for you for getting maximum score, the CBSE board exam. The tips for the exam, we always need to compare the DNA and RNA structure. The DNA RNA structure, stability, mutation rate, and roles in the gene expression, everything we need to understand in detail. So one is chemical structure, another is stability, another is mutation, another is genetic expression. All these four are matters. So can you say the gene expression of the RNA? Yes. The RNA, when it is getting expressed, it makes protein. When the RNA is getting expressed, it makes DNA only in the retrovirus. Can you explain the mutation possibility in the RNA and DNA? RNA is highly prone for getting mutated than the DNA. So then if you're coming for the next segment is the chemical structure of the DNA. If you are talking about the difference between the DNA and RNA is basically the uracil and the thymine. Uracil and the thymine. These are all the messages we should understand when you are studying the tips for the exam.